It's great that we have Liam Paninski to give the next lecture. Liam has done a lot of very interesting and important work at the interface of statistics, neural data analysis, and he did his PhD with Eros Simoncelli, I think, at NYU. And then he's been a professor at Columbia for many years now. And I've always seen Liam as making all the great technical advances in neural data analysis. And today he's going to tell us about some of those, I hope. And I think that we should all carefully listen and learn a lot of the stuff from him. Thanks, Liam, for accepting to be here.
All right. It's a pleasure. I wish I could be there in person. The photos look pretty amazing. Maybe in future years.
Yeah.
So cool. Let me share my screen. Oops. Host disabled participant screen sharing. Can we fix that?
Yeah, we're working on it.
All right, cool. Yeah, so there's some construction going on here, so apologies in advance if you hear some banging. I've put myself downstairs in my daughter's bedroom to hide out a little bit, so hopefully it won't be too noisy here. All right, looks good. All right, so I'll share my screen. I'll do this. All right, does that look okay?
Yes.
Yeah, all right, awesome. Cool. So as always, please interrupt me as much as possible. I'd love to, I feel lonely here talking into the void if I don't get any questions. So, I think I've got, plenty of material to discuss here. And if we don't get to all of it, that's totally great. I'd love to just hear as many questions as you have. On the other hand, I can't see you very well. So, yeah, just shout at me if you want me to stop and talk about anything that's a little bit more. Okay. Yeah, this talk, I'm going to go over some of the kind of things I see as important in kind of modern neural data analysis. A lot of these topics were actually described in a review article that I wrote with my friend John Cunningham that came out a couple years ago. Everything we said in that article is still pretty current. So take a look at that if you want to kind of look a bit more in the literature and kind of get into these details a little bit more. All right, cool. I'll jump right in. All right, so yeah, I mean, this is a really exciting area to be working in right now. So I think there's just an incredible amount of cool stuff to do in this area, right? So we've got fast, big parallel computation, right? We've got, obviously there's been an explosion of great new machine learning tools over the past decade that's changed the world, not just neuroscience. We have these science fiction methods for recording from large-scale populations of neurons, even whole brains in some contexts, and we can also kind of shoot laser beams into the brain and affect behavior. Right? It's kind of a crazy science fiction world we live in, and kind of more importantly, I see a lot of the the critical open questions in neuroscience, the fundamental problems that got a lot of us into neuroscience. A lot of these are actually kind of statistics problems if you look at them the right way, and if you kind of translate these neuroscience problems into statistics or machine learning problems, you can make some really kind of nice progress. One of the big bottlenecks I see in the field is that we just don't have enough people who can kind of speak both languages of statistics and machine learning and neuroscience and kind of translate back and forth and push this field forward. So I hope that some of you kind of, get into this area and help us kind of solve some of these really exciting problems. So what do I mean by neural data science or statistical neuroscience, right? Maybe unsurprisingly, I try to take a pretty broad view, a greedy view of what statistics can kind of bring to neuroscience, right? So some of these kind of statistical neuroscience questions are very classical, right? We think about encoding problems and decoding problems, right? What information is encoded in large populations of neural activity? How do we decode that information into things that are useful? Obviously, there's been some pretty cool YouTube videos on the decoding problem recently that's driven a lot of interest in this field. Another big question, can we take populations of activity, can we look at hundreds of neurons kind of firing away and figure out how things are connected to each other, right? That's a topic that I know has been interesting to some of the speakers in this summer school. Or vice versa. Can we, if you hand me kind of an EM reconstruction of a brain or a small part of a brain, can you predict what the dynamics of that circuit are going to be, right? So we can go kind of back and forth here. Now that we have the technology to record from multiple regions of the brain simultaneously, one of the really interesting questions these days is to figure out, how do the dynamics of one brain area impact the dynamics of another brain area, and what kind of cool circuit models can we build that are constrained by data to kind of make inferences about that. I already mentioned, optogenetics, and then of course you can also perturb the brain with magnetic or electrical kind of stimuli. So that brings up a really interesting kind of real-time optimal control problem, right? Can I kind of record some activity, figure out what's going on in that circuit, and then kind of optimally perturb that circuit to behave in a way that I want it to behave, or to optimally figure out what's connected to what, for example. And then there are more basic questions. What is a cell type, right? How do I determine cell types from morphology and molecular information and functional information? How do I figure out how many cell types there are, right? These are just completely fundamental neuroscience problems, but again, they're really kind of statistics problems in disguise. What is the best experiment I should run next? Again, just a completely fundamental problem that experimentalists have to answer every day. And if you bring to bear the technology of optimal experimental design, you can kind of do interesting, cool things. I could go on, right? So there's lots of really fundamental open neuroscience problems that are really statistical in nature, right? So those are the kinds of problems we want to tackle. One of the big motivations for my group over the last few years has been this collaboration we're a part of, this International Brain Lab. So if you haven't heard of this, you can check out this kind of position paper that we published a couple years ago, and then more recently we have kind of our first big kind of data release that happened, I guess, this year. So the point of this lab is to try to record the electrical activity from the whole mouse brain as the mouse performs a kind of a well-defined simple behavior, right? So we want to understand decision-related activity throughout the whole mouse brain. And the way we're attacking this is we have about 10 or so experimental labs who have set up this behavior that's described in this paper, kind of tried to standardize that as much as possible across the 10 different experimental labs. And now we're recording with neuropixels arrays in those mice as they perform this task. And we're trying to kind of poke around in the brain, taking hundreds of experiments. So we really try to cover the whole brain, and so we can record the activity in the mouse brain as these mice perform this well standardized and very reproducible decision task. All right, and so. So that's like the job of the 10 experimental labs. And then we also have 10 theory or analysis labs who are, trying to make sense of all this data, right? So this is a huge amount of data. It's a huge effort. And here's kind of what things look like ideally, right? So we have this experimental data that are being kind of recorded. I mentioned the neuropixels recordings. We're also doing some 2-photon recordings and some fiber photometry recordings. We're also recording behavioral video in the animals that are performing this task. And basically all of this, big data is being processed and put up into the cloud for everyone to use and take a look at. And then of course, the analysis labs and the theory labs have all their theories of what the brain might be doing, and we're trying to kind of connect those theories to the real data. And then, of course, suggest new experiments that can be done to pursue specific experimental questions that are related to these theories. All right. So like I said, some of this data is already available. We already have a bunch of behavior data that you can download. And we've also released a tiny bit of ePhys data as well, just the recordings from, I think, just 4 or so mice so far. We're still in the thick of recording and kind of quality controlling all these ePhys data sets, but more of that should become available over the next year or so.
Yeah, can I?
Yeah, go ahead.
The experiments, are they distributed between different labs depending on the modality? So some labs are doing calcium imaging, some are doing electrophysiology, or is it also mixed? Yeah,
great question. So we kind of have platform papers and individual projects that are done in these labs, right? So the first platform paper was this behavior paper where the 11 experimental labs got this behavior working kind of reproducibly across all the labs and did some quantification of what the mice are actually doing as part of this task. The second platform paper is going to be this ePhys paper, where we release the recordings of these neuropixels recordings across the whole brain. But then in parallel, some of the labs are pursuing these fibrophotometry experiments to look at specific questions. Some are doing wide field calcium imaging. Some are doing 2-photon calcium imaging. So those, all of the data will be released, but the large scale platform papers we're starting with are based on neuropixels ePhys recordings.
And following up on that, so the neuropixels, how stable are they in mice now? I think they're quite stable in mice, right? But in rats not so much, or is it the other way around?
Yeah. I'll get to that. One of the big kind of meta messages of this talk is going to be to never trust your data. So, one of the bitter lessons I've learned over my 2 decades or so in neuroscience is you can make beautiful theoretical models, and when you actually try to really closely connect those to data, you start to see issues, right? And you need to be extremely careful about the data quality as you're thinking about what models you can build, what models you can test. So that's going to be something that I'll come back to in the context of all these different data types. But the short answer is you can definitely do cool things with current neuropixels recordings, but you also need to be careful and check the data very carefully. And I'll talk about some of our experience with that so far. Any other questions? All right, cool. Yeah, like I said, so like I said, you can go to this website, take a look at the data, and yeah, we would consider it a failure if we release this cool data and no one publishes any papers to using this data. So we hope to get as many people using this data as possible and looking at it in a lot of detail and discovering flaws in the data and using the data to test their own ideas. And again, to suggest new experiments, right? There's lots of, we hope that this is kind of a platform that can be built upon in the coming years to do new cool scientific work. Okay, so like I said, this has really been a key motivator. Another big issue that we've kind of run into as part of the IBL, but also just more generally, is the difficulties of So my group and a lot of other groups obviously are in the business of developing cool new analysis methods, cool new machine learning methods that can be applied to neural data. And what we've discovered over the years is that there's different levels to this, right? You can get your cool new method working on one data set and publish a paper based on that. That's a good new step. It turns out to be a lot harder to actually make tools that other people can use reliably. And it turns out to be even harder to make tools that kind of scale not to a single data set, but to multiple data sets, right? Of the scale of the IBL, for example. And I think there are a lot of things going on there, right? One of the basic problems that we have when we release new analysis tools is people find it difficult just to sometimes install the tools on their own local machines, or get things to run, or have enough memory on their machines to run things. And there's a huge amount of kind of time spent on just getting these basic methods working. And so we kind of, half jokingly refer to this as infrastructure as grad student, right? I'm sure you all know, you've probably spent a lot of time, trying to get other people's MATLAB code to run, or Python code, or installing different Python environments, installing, maybe getting GPUs to work nicely on your local machine. It's a lot of work, and we've realized that a lot of this work is kind of wasted, and it's 2021. There should be a better way. So again, this work is in collaboration with John Cunningham's group. There's a bioRxiv paper on this work. What we've tried to set up is a method where experimentalists can come with their data, not have to install anything in Python or on CUDA or anything. They just upload their data to the cloud, along with some configuration files, and then this platform, this neuroscience cloud analysis platform, basically kicks off a few instances on AWS to process your data in parallel, and then it returns the results and kind of a certificate of what was actually done. So the analysis is kind of completely reproducible. And so this is set up. You can use it. And I think this approach has a lot of advantages, right? It's infinitely scalable. If you have 100 data sets you want to process, you can just upload those all and kind of process them in parallel. You can run as many different kind of settings of your hyperparameters and do hyperparameter search at scale. You don't have to mess around with CUDA at all. You don't have to install anything. And actually, it turns out to be kind of faster and cheaper to run things in the cloud than it is to run things locally, typically, unless you're really running things kind of around the clock on your local machine. Because essentially with this cloud platform, you just rent hardware for just the amount of time you need to run your analysis. So again, this is kind of a new tool that we're hoping that everyone starts to use. We have a bunch of kind of different methods that are available now. You can check out the preprint on bioRxiv for more details. And for people like this crowd who are interested in developing new analysis tools and new theories, we also hope that you can kind of bring in your own kind of theoretical tools and analysis tools and make those available to everyone as part of this platform. So we've tried to make it easy, not just for users to use the system, but also for developers. And again, the advantage here is that what we found is that if you can get your code to work nicely and kind of optimally on one AWS machine instance, then you don't have to support Windows and OS X and all the other nice operating systems that we have. You just have to get it working well on kind of one machine, and then you're good to go. So that makes life easy on the developer side as well. Any questions on this one?
Liam,
Yeah.
how do you deal with the problem of having basically as many data formats as there are experimental labs globally?
Yeah, that's right. So we've kind of punted on that. So again, we set up this platform. You can upload data of any type to the platform. But if you want to use one of these methods, like if you want to use deep lab cut, for example, each of these methods is going to accept some format, right? So if you want to use this method, you might have to record data in that format, or write a little wrapper file to get your data into that format, and then you're good to go, right? So some of these methods are going to accept multiple formats, some of them are not. But we're not in the business of enforcing different formats. We kind of leave that to the developers of these methods to handle that, and that actually hasn't been much of an issue for us so far. Maybe it will be in the future, but for now that hasn't been the bottleneck. Any other questions? Okay, cool. Great. So let's get to some science, right? So one of the big out of the way. One of the big things that kind of motivated our early work in calcium imaging analysis was this question of, calcium imaging, became like a cool thing maybe a little more than a decade ago. And immediately we're like, okay, now we can see the nice twinkling of lots of cells simultaneously. Maybe we can look at this large scale neural activity and figure out what's connected to what, right? Can we do circuit inference from functional data? And so what are the problems you would need to solve to make this kind of a reality? Well, you would need to take the raw data and figure out where all the cells are. If you have any overlapping cells, you're going to need to kind of demix the activity of two different cells that lie on top of each other. You're going to need to kind of extract, we all know that the calcium imaging is fairly slow, right? Every time the neuron spikes, there's, calcium comes into the cell and then slowly kind of decays back down to some baseline. So you're going to need to run some deconvolution method and maybe a denoising method to pull out neural activity or try to estimate neural activity from that the slow fluorescence kind of recordings you take. There's been a huge amount of work in trying to go from lots of spike trains to kind of a circuit model. That's actually something I'm not going to talk about much today, but I think you've heard some of these ideas in some of your other lectures. So you want to take these spike trains and go to something that looks like this, kind of a directed circuit model. And then maybe if you're lucky, you can hook up with an experimentalist who's willing to start to poke these cells with lasers, right? Maybe if you think this cell is connected to this cell, you can actually optogenetically stimulate this cell and see if you get a bump of activity in the nearby cell. And then hopefully kind of close the loop and kind of collect more data and do this all again, right? So this was kind of the dream experiment that kind of motivated our work for a long time. And I still think it's kind of a cool thing to pursue, but one of the things we realized over time is that it's really hard. There's a lot of things that have to kind of go right for this to work, and this is really one of the cases where we learned early on that you really had to pay very close attention to your data quality, or else your results are just kind of very hard to interpret, right? So for example, if you don't solve this demixing problem correctly, if you confuse the activity of one cell with another, you're going to get kind of mistakes in these arrows here, right? That's pretty clear. So we ended up spending a lot of time focusing on this demixing problem. Another issue is, maybe the activity in this population is kind of non-stationary, right? So maybe, how do you deal with non-stationarities or changes in the SNR of this population over time? What do you do if you're only recording a small bit of the circuit and there's lots of common input coming in from the rest of the circuit, right? So there are a lot of, again, I want to emphasize this. You want to make sure that the theories and the computational methods you're working with are you want these to be as connected as possible to the real data, and that's a lesson we've learned over the years. So let's take a look at what some of this data actually look like. So I don't know if you've talked much about calcium imaging data before, if you've seen calcium imaging data, but here's an example of what some raw data look like. All right, so this is a raw video. This is actually taken from Andreas Tolius's lab a few years ago, just to give you a sense of what this looks like. Right, so you have a few different components in these videos typically. So each of these little blips here corresponds to the activity of one cell. If you zoom in on this, you can kind of see the soma kind of very clearly outlined by the flashes of this activity. You also see a lot of kind of fuzzy noise. There's also sometimes some kind of background activity where it looks like a lot of the image is kind of coming up and down together. And you want to kind of build that into your models, right? So the basic problem we want to solve here is to separate all these different aspects of the movie apart. If we think of the video as a matrix, we're doing matrix factorization, right? So to be clear, the matrix that I mean here could be kind of x by y by t, or if we kind of vectorize each frame of this video into a single, d by t matrix. D is the total number of pixels in the video, and t is the number of time steps. So we want to take that d by t matrix and decompose it into a noise component, maybe a background component, and then also individual components that correspond to the activity of each of these individual cells that we see, right? So that's kind of what I'm showing over here. We're trying to go from this raw video to ideally something that looks like this, something where we've separated out the background activity, separated out the noise activity, and now we just have the activity of each of these individual cells hopefully is demixed and localized as well as possible. Any questions on the basic goals here? All right, so I think a lot of you have probably seen examples of this, or maybe even used some of these methods on your own data. So what are the what do we actually do with this data? Well, like I said, the kind of dominant paradigm in this field is this kind of matrix factorization, right? So if I take that video that I showed you on the last slide and kind of break it up, so we vectorize each voxel or each pixel. So we have d pixels down here and t time steps over here. What we end up with, this is simulated data to be clear, but real data looks similar, just noisier. We see something like this, right? So every kind of stripe here corresponds to a single cell firing. Each cell covers multiple pixels. It jumps up, the calcium level kind of jumps up pretty quickly and then decays down slowly over time. And that's what we're seeing here. And you might have multiple cells kind of overlapping spatially. Maybe one cell is slightly behind another in space. And when you scan your laser across this field of view, the activity of those two cells might be combined in a few pixels. And so that's what's being shown right here, right? You have a couple cells that kind of overlap spatially, and we want to demix the activity of those two cells, right? All right, so you have this data matrix Y, this d by t data matrix, and we want to separate this out into lots of other more interpretable matrices, right? We want to separate out the noise and the background and the activity of each of these cells, right? So this y hat is what I'm going to think of as this cleaned, this denoise video, right? And that is going to be decomposed into smaller components as well, right? So basically what we model here is that this denoise component of this video corresponds to the sum over all the neurons that are visible in this field of view, right? So R here, this R corresponds to the number of neurons that are visible. AI of X is the spatial footprint of each of those cells, right? So I indexes cells here. This is a purely spatial term. So this corresponds to kind of the spatial footprint of where the cell is in space. CI of T is a purely temporal term that says, okay, how is this cell's fluorescence, overall fluorescence, kind of jumping up and decaying over time, right? So we want to extract all of these spatial footprints and all of these temporal terms from the video. And then, of course, we know a lot about these temporal terms. We know they kind of jump up and decay down and jump up and decay down. So we might have an additional kind of model for these temporal terms that corresponds to kind of a sparse spike train that's being convolved with kind of maybe an exponential filter or something like that, right? So if you write out everything we know about this data, or all the kind of the prior knowledge that we have, there's a huge number of constraints that we can impose on this matrix factorization problem, and use those constraints to kind of get better recovery. And we've developed some methods based on this constrained non-negative matrix factorization model. A lot of other people have developed good methods that are kind of variants of this, right? So maybe you have a different way of initializing these different terms, or maybe you have a different noise model, or maybe you have a different kind of model for this kind of large scale background fluctuations. But a lot of the methods that you've probably seen in the literature are kind of based on this, variants of this matrix factorization approach, right? Any questions on this? Comments?
Yeah, I'm just wondering how you distinguish the background activity versus the noise activity. Is it based on the frequency of the
That's right. Yeah. So let me go back to that video. I actually used to have some slides on this, but well, you can see another example here. Right. So this is another raw video right here. You can see, lots of stuff going on. We've kind of zoomed in here a little bit more on the individual cells. You can see these individual cells a little more clearly. You can see a lot of shot noise. You also see these stripes that sometimes come in as a result of motion correction of the data. And if you watch this data long enough, you can kind of see these kind of fluctuations of the background, which we believe is probably a result of axonal input coming in and hitting lots of these cells simultaneously. And the axons are so small that we can't actually, see their shapes at the resolution of the 2-photon microscope, at the resolution these data are taken at. And so this is kind of an example of this background activity that we've tried to extract. There's a lot of stuff kind of going on in the background, but we try to distinguish that from the activity of these individual cells. So yeah, the question is, how do we actually do that? It's a little bit heuristic, right? These background components are going to be the sum of the activity of multiple cells typically, or kind of a widespread array of axons coming in and hitting this little population of cells. People have tried different things. As you said, maybe there's a spatial frequency dependence of the background that's different from the individual components. Obviously, individual components tend to be much more localized than this kind of large scale background activity. There are a few kind of tricks that people have used to try to get at this. But I think there's actually more work that could be done there to really nail that down. So that's a great question. I should also say that there's lots of different data types available. I'm gonna show a couple. This is kind of standard 2-photon data. That's where you zoom in. Typically in a head fixed mouse, and you can zoom in and look at these cells in great detail. There's also 1-photon imaging, where you stick a little camera on a mouse's head and let it run around. That data looks very different from this 2-photon data. I'm not going to get into the details of that in this talk, but it turns out that you need a wildly different background model for that 1P data than you do for the 2P data, just because of the details of the imaging and the way that the fluorescence actually gets onto your imaging apparatus is pretty different. So you do need a very different model for that data. We'll see some data in a little while that corresponds to voltage imaging or dendritic imaging, and you need different kind of models for those types of data. So that's a great question. Any other comments or thoughts? Okay. All right. So like I said, we've been trying to develop and kind of optimize these matrix factorization methods for a long time. One of the things you should know about non-negative matrix factorization is that it turns out to be an NP hard problem, which is kind of bad news. So like I said, all of these different methods try to use different heuristics based on what we know about the real data to kind of initialize these methods well and kind of pull out a good solution. What we realized a couple years ago is that a simpler approach, thinking about easier mathematical problems that we can apply to matrix factorization. A simpler approach is based on PCA, right? So we know that PCA is not an NP-hard problem. You can just run PCA on whatever data you like and get a nice answer pretty quickly. What PCA does is it's not going to give you the individual components of these individual neurons, but what it is very good at is kind of compressing your data into a lower dimensional space where the search for these components is actually going to be much faster, right? So that's something else we've worked on in the past couple of years. We've developed kind of a a souped up version of PCA. So it turns out that applying PCA to the raw data is probably not exactly what you want to do. It doesn't take advantage of everything we know about these signals. But if you apply kind of a penalized version of PCA locally in each little block of your data, you're able to actually get a very good compression and denoising of the data, which is shown right here. Okay, so we've developed this kind of penalized matrix decomposition. Think of it as just a fancier version of PCA, which is more matched to the data properties here. And if you apply that penalized matrix decomposition to this raw data, you get something like this. And the great thing about this data, you can see it's much cleaner than this data. It's also much smaller, right? So this data lives in a subspace which is 100 times smaller than the raw data. And what that means is that you can pass this data back and forth more easily to your friends. You can share data. You can store data much more cheaply, and you can actually run these fancier NMF methods much faster on this compressed data. So typically, even large data sets will now fit in GPU, and now you can run your favorite demixing algorithms very, very quickly on this compressed data. So that's kind of an important kind of trick we've learned over the years. If you can take this raw data and compress it into a much lower dimensional feature representation, you can kind of do cool new things with the data. So this is an example of that. Any questions on this, or should I go ahead? All right, cool. All right, so let's go ahead. I mentioned, there's lots of different types of imaging data these days. This is a beautiful example from the Hillman lab at Columbia, where they're actually imaging in 3D, not just 2D. And what we're looking at here is the raw 3D data just max projected into 2 different dimensions, right? So we're looking at a little 3D volume of cortex. Here's kind of the top view of that bunch of cortex, and really layers. Really layer 1 of the cortex, looking down upon it. Here's a side view where we've projected kind of into the side. So you see all these beautiful apical dendrites kind of coming in from the bottom of the brain here up to the top of the brain. And again, the game is to try to take this raw 3D data and decompose it into background and noise and the individual kind of neural components. And so a lot of these NMF methods that I was discussing on the last slides can be adapted to this kind of data. This is just, I find this video very calming and nice to look at. It's like watching a fireworks display.
Excellent. The colors to what are referring to, the mapping of the color with the activity.
Sorry, I didn't hear that. Can you repeat that?
Mapping of the colors with the activity in the 2 central.
Yeah, so I think the question was about how do these colors correspond to what's going on in the raw data? So all we've done here is we've just assigned a different color to each of the components that we've pulled out of the data. So we've pulled a few hundred components, these A times C components out of the data that hopefully correspond to single cells, and we just assigned a different color to each of those components, just so we can visualize the activity here.
Thanks.
Any other questions? All right, awesome. Okay, so I've been talking about calcium imaging data so far. The other big advance that's happened, over the last decade, but is really accelerated over the last couple of years is the technology for imaging voltage directly in the brain, right? So again, just a brief reminder of the biophysics here. Calcium imaging doesn't image neural activity directly. Instead, you get this kind of indirect readout of activity based on the influx of calcium and then the kind of slow extrusion of calcium in individual cells. So the advantage is it's a nice bright signal that you can pull out. You can image lots of cells simultaneously. You can label these cells molecularly so you know what the different cell types are. You can image these cells over multiple days in a lot of cases. So calcium imaging is a great technique, but it's limited in that it doesn't image the raw voltage of these cells directly. So what people have been working on is this kind of very clever, very hard protein imaging protein engineering to develop proteins that will be inserted into the cell membrane now instead of living within the cytosol. You put these proteins into the cell membrane, and then the hope is that these proteins will change their fluorescence based on the transmembrane voltages, right? So these are direct readouts of the voltages of these cells. Sorry, say that again?
Yeah, isn't that going to affect the properties of the neurons you're studying?
Oh yeah, yeah. This is kind of the bleeding edge, and so yeah, maybe if you're not careful, if you pump too many of these proteins into the membrane, maybe you're going to change the electrical properties of the cell. Maybe you're going to change the health of the cell overall. There's all kinds of issues here that again you need to be pretty careful about. And so if you read these voltage imaging cells, you know, typically the last figure in the paper is always like a beautiful image of the voltage imaging that you can do, but the first 5 figures are all about, well, here's how this new protein is expressed on the cell. Here's a figure showing that I don't think I hurt the health of the cell. Here's an image showing, you know, kind of ground truth comparisons of the, you know, electrical activity recorded, you know, intracellularly versus the voltage readout that I get from these images. So yeah, it's very hard work, and you definitely want to be cautious about that. That's a great point. Another big issue, well, there are a lot of issues with this data. So it's targeting what we want. It's targeting the language of these cells, the raw voltage on these cells. But there are a couple disadvantages, right? First, you have to image much faster, right? So voltage fluctuations are on the order of milliseconds, whereas calcium fluctuations are on the order of hundreds of milliseconds. So to capture these voltage signals, you have to image about 100 times faster. So this data that I'm showing here, which is from a paper from a couple years ago now, is taken at 1 kilohertz, right? So you have to have very fast cameras. The data set sizes explode. So that's one issue. Another issue is that because the voltage sensing proteins are targeted to the kind of 2-dimensional surface of the cell rather than the 3-dimensional volume within the cell, it turns out that they're just basic kind of, you know, the voltage sensors are going to be lower SNR than the calcium sensors for the foreseeable future, right? You just have to accept that these signals are much noisier than you might be used to seeing with calcium signals. So that's shown up here. Okay, this is actually the raw data taken from one of these experiments. And if you stare at these cells for a long time, you don't see anything at all. You really need to do some processing to start to see the fluctuations of voltage in these cells, just because the SNR is much lower. So what we did to go from here to here is simply kind of extract an estimate of the baseline fluorescence. Basically just extract out the local mean from the left panel to get to this. And now you can start to see some quick flashes of activity, but they're very, very noisy, right? So again, it's pretty critical with this voltage data to apply some kind of denoiser to it, so that you can actually start to see the voltage fluctuations, and that's what we're showing here, right? So this is this penalized matrix decomposition applied to this you know, detrended raw data, and now finally you can start to see things that are going on here, all right? And what you see if you start to stare at these data is a few different artifacts. So you don't just see the nice flashing of individual cells. You actually can see kind of the motion of, you know, as this mouse moves around, you start to see kind of the little fluctuations and kind of the shape of these cells, which you don't want to see if you're kind of if you have kind of a well-stabilized prep. So you have to deal with motion artifacts. That's very important. You also see, if you look at this panel, you can see it very clearly. You see these little diagonal stripes in your data, and you know what that is. You might think, oh, that's a cool kind of spike traveling along the dendritic tree, and I'm going to be able to study back propagation, and that's going to be really cool. That's not what this is. These are actually blood vessels, right? So what we're seeing here is actually the flow of individual very small blood cells flowing past your field of view. And if you don't handle that that contamination properly, it's going to completely confound any downstream analysis you want to do of the subthreshold fluctuations. And maybe you want to understand how this spike over here affects this cell over here. You need to be very careful about these artifacts in the data before you get to those kinds of analyses. And again, these are things that you just wouldn't even be able to see in the raw data. You need to perform some kind of denoising before you can even see these artifacts in your data. So it's very important to be careful here. All right. So if you're careful about that, if you kind of mask out these blood signals, and if you regress out these motion signals, finally you can get to something that kind of hopefully is close to kind of a simultaneous subthreshold recording of multiple cells simultaneously in vivo. So now the hope is that we can start to do some cool science with these methods, and some of that is kind of described in these papers that have started to come out from multiple labs now. Any questions on this?
So on the graph on the right, are you able to define spikes, or is it simply subthreshold fluctuation?
I'm sorry, the audio is pretty tough. So you asked a question about what's going on the right?
Yeah, are you able to classify spikes in this, or these are all subthreshold?
Yeah, that's right. Okay, yeah, I didn't kind of walk through the full story here. So we perform these different kind of denoising steps, and then finally we run this kind of matrix factorization step to pull out the individual activity of these cells. And so what's being shown over here, this is the spatial footprint of this cell down here. And this is the temporal kind of trace that we pull out corresponding to this cell. All right, so you see some kind of subthreshold stuff, and occasionally you see some kind of spiky things. And then the question is, okay, is that a spike, or is that just kind of an artifact? And you need to be careful about that. Here's another one where you can kind of very clearly see a little bit of axon and a little bit of dendrite coming off of the cell. And these spikes, if you kind of stare at this video and go back and forth and look at what the cell is actually doing there, you can kind of convince yourself that these blips actually correspond to spikes probably. And so you can look at each of these components individually and go back and forth. And we call this a demixing video, where we've tried to decompose the raw data into these different components, and then try to be careful and convince yourself that what you're seeing is real and not just some kind of artifact of demixing or some other thing that's going on in your data. And so yeah, you have to be very careful about kind of performing these visualizations and checking to make sure that you think that what's going on here is kind of correct. But there's no ground truth for these data sets. It's not like we could stick 5 electrodes into these cells and check that they're all correct. You can stick kind of 1 electrode at a time in and try to make sure that you're getting kind of the correct signals out. But it's impossible to kind of ground truth at scale for these videos. Did that address your question a little bit?
Yeah, also, for example, the motion graph, it seems very ambiguous when the spikes and when it's just fluctuations. So,
yeah, that's right. So for some of these cells, it's hard to tell what's super threshold and what's subthreshold, right? So that's just we've tried as hard as we could. The protein engineers and the physiologists and kind of the data analysis working together have this is as good as we've gotten this cell to be at this point. But I will say the technology is marching forward, right? People are every few months there's kind of a new iteration on these voltage sensors, so it's definitely getting better. So if you look at something like this, and again this is a couple years ago now, if you look at something like this and say, well, that's not the SNR that I want to be dealing with, check back in a few months or a year or two, and hopefully these spikes will get higher and higher over time. So that's definitely a work in progress. Any other questions? Okay. So that's kind of where things are in the voltage imaging world. One more thing I wanted to kind of discuss before we move this calcium imaging stuff is I guess I need to speed things up a little bit. That I mentioned this question of ground truthing, right? So how do you know if the signals that you're extracting and the shapes that you're extracting actually correspond to kind of the real underlying kind of microanatomy of these cells? So there's been a lot of effort there, but it's been pretty unsatisfying. So people have taken videos and then kind of hand labeled cells in their videos, and then tried to compare those hand labeled cells to the output of these different algorithms. I think that's a good step, but it's clearly incomplete because we know that two different hand labelers will kind of label different cells, right? And these hand labelers tend to be biased towards somas, and they kind of miss all the interesting little dendritic bits that you might see in these videos. So that kind of hand labeling work has always been a little bit unsatisfying. Something else you can do is shown here. What you can do is record a calcium imaging movie in a mouse, and then take that volume of mouse brain and slice it up really, really thin, put it through an electron microscope, and then run, your favorite modern segmenter on that EM data to pull out individual cells, and then try to map those individual EM cells back onto the functional 2P data stack that you collected, and then try to check your results that way, right? So that gets us closer to what I think of as a, I don't want to call it ground truth, but it's closer to a gold standard that we can kind of really objectively kind of rank these different algorithms on. The other thing you can do, of course, is try to actually use these shapes that you've extracted from the EM data, use those as priors for what your cells should look like, and then try to extract even cleaner representations of the neural activity using those strong prior information about where the cells are. Okay, so this, I'm summarizing kind of a large multi-year collaboration that we've just played a small part in. So my lab has been involved in trying to make this connection between the segmented EM data, which is an extremely hard problem that's been kind of represents a decade's worth of work in that community. Take those beautiful EM segmented cells and match them and use them as priors to extract even cleaner calcium signals from these brains. Okay. And again, you can check out the details of that. Again, it's all based on this matrix factorization approach, but using the EM information as priors on these spatial components that we're going to pull out of this data. Okay, so if you run that analysis, you get this beautiful. This always kind of chokes my poor little laptop here, but you can see basically simultaneously the activity of this small neural circuit along with the full kind of microanatomy, right? So you can see where all these cells are. You can actually see which cells are connected to each other, and you can map that calcium imaging. Those flashes of activity that you get from the calcium imaging data, you can map that back onto this microcircuit to really connect the function of the circuit to the structure. So I think there was just another big data release from this collaboration. Again, if you check out micronsexplorer.org, you can get this raw data, and there's a huge number of cool scientific questions to be addressed here. So I think that's going to open up a lot of cool new work. Just to close the loop on that, to give you a sense of what these cells actually look like as you translate between the EM and the 2P imaging. In this case, the experimentalist imaged kind of across multiple sessions. They took multiple planes of 2P data from this brain volume. And then our job, like I said, is to kind of extract those AIs, those spatial footprints, and then try to map those back onto the underlying kind of micronet, right? So here I'm showing the segmented cell, a single segmented cell out of hundreds out of this little volume. I should say hundreds of cells that include somas. There's actually hundreds of thousands of little axonal bits that course through this small volume. And then what we're trying to do here again is we start with the EM mesh, then we say, okay, if I were to use a 2-photon imaging microscope to take a little slice through this data, what would this EM mesh actually look like? So we can kind of generate kind of predictions of, if this cell was completely filled with calcium indicator in a uniform way, this is what, this image is what a single slice through that cell would look like. And then if we compare this kind of predicted image to this, what we get out of the actual calcium imaging data, what we see is, as desired, this footprint lives within this cell, right? There's no leakage of calcium indicator outside of this cell. That's good. But it turns out that this this calcium footprint always lies within kind of a strict subset of the potential volume of the cell. So basically the bright parts of the cell are just going to be kind of a subset of what's predicted by this EM mesh. And if we collect that information across multiple cells recorded in this volume, again, we have this kind of gold standard data set of these calcium shapes now, right? So the brightest 10 calcium shapes shown on 3 of these slices are shown here. The next 20 are shown here. The next 30 are shown here, and so finally you get down to this really small stuff that you probably wouldn't have been able to pull out if you didn't have strong prior information about where these dendrites were kind of coursing through the imaging planes here. And the interesting thing to me here is that these somas, these big bright somas that people were picking out in these kind of human labeled data sets really comprise just about 20% of the components that you can pull out of this data. There's a huge amount of signal that you can extract beyond just these big bright somas. So that's kind of an important kind of take home message here. And now, of course, now that you have these gold standard kind of cell shapes, you can use those to train fancier denoising methods, fancier methods for demixing very, very dense data. There's a lot of cool new things we can do now downstream. Okay.
And did as you're assigning it to the somas and the axons and the different parts of the EM reconstruction, does it fit with what you'd expect, like that the activity follows?
Yeah, that's right. So that's kind of a big part. Oops. That's kind of a big part of this paper. Trying to convince ourselves and kick the tires of this method and kind of establish that these footprints that we're pulling out of the data kind of match the EM priors very well. And again, to be clear, in this case they have to, right? We've constrained the matrix factorization so that so again, each of these footprints actually live within the predicted EM footprints, right? Because in this case we're interested in trying to extract out the temporal activity of these cells as accurately as possible. What we haven't done much of actually, and what we should do more of is take the different algorithms that are out there, run them kind of just directly, and then go ahead and try to match things back to these ground truth or these gold standard 2P footprints. That's something we haven't had time to do, but I think would be a great project to pursue in the future. Any other questions? Okay, awesome. So that was a lot. We just spent a lot of time talking about voltage data and calcium data and this new EM kind of calcium imaging data. I should say that, what I wanted to do in this talk is talk about kind of three types of data. There was the imaging data that we just spent a lot of time on. I wanted to talk about behavioral video data. And I also wanted to talk about electrophysiology data, these neuropixel recordings. I was targeting an hour and a half. Yeah, maybe I'll just go ahead. And if you guys get bored or if you want to hear one thing or another, just tell me to speed up, and I'm happy to kind of adjust my pace as I go. All right. So I guess the second big kind of type of data that we've been really occupied with over the last few years is this behavioral video data. It's become very clear that if you go back to that first IBL paper that I mentioned, the behavior paper, where we kind of take this fixed behavior across multiple experimental labs and try to compare the behavior of multiple mice. That whole paper was really based on very simple readouts of the mouse's behavior, right? So in this IBL task, you see this head fixed mouse. It has to this is a little Lego wheel. It has to roll this Lego wheel back and forth to kind of make a decision based on what it's seeing on the screen. So it's playing a little visual video game, and its behavior on each trial can be seen as just a simple binary thing. Should I go left or should I go right? right? It's a very simple kind of 2 alternative force choice task. And so if you just look at that single binary output per trial, you can do a lot, right? You can make a lot of cool models based on that binary output. But if you look at what the mouse is actually doing, there's actually a huge amount of stuff that's going on that's not just directly related to this binary output, or that might be correlated to this binary output or to the mouse's priors about what's going on in this task. But there's obviously a huge, rich set of information that is going to be driving lots of neurons in this mouse's brain. And if we throw this information away, we're gonna miss out on a lot of cool stuff that's going on in the brain, right? And it's become very clear that these little movements of the mouse's whiskers and the licking and the little kind of movements of the paw that it's doing have a huge impact, kind of an outsized impact on the activity of neurons throughout the brain, not just in kind of the motor areas of the brain, but also in the visual areas and the auditory areas, and really throughout the brain. And so we want to extract as much information from these videos as possible. And there's been some really nice work on this problem over the last few years, right? So probably a lot of you have heard of deep lab cut or have used this method. Basically, what you do is you take some video, you add a few labels to the mouse's paw or the pupil or the nose. You train a little neural network that kind of tries to identify those objects from video, and then you throw this at a lot of test frames video, and you're able to kind of track the mouse's behavior over time, right? So that's a beautiful piece of computer vision technology that wasn't available 5 years ago, and it's made a huge difference in the way we approach a lot of questions in neuroscience. So that's been extremely impactful. So we've been using Deep Lab Cut as part of the IBL. And what we've realized is that, as you go again from the activities, the recordings in a single lab to the recordings, to hundreds of sessions across a dozen or so labs, it turns out that methods that work really nicely in one session don't work at all on multiple sessions. So this has been kind of another bitter lesson that we've learned, right? So you actually have to work very hard to get these methods to work at scale. And so we've been interested in ways of improving these methods and scaling them up. I mentioned this Neurocast platform. That's been a big part of how we kind of approach these problems these days. So you can see what's going on here. This is after actually labeling more than 10,000 frames, so a pretty large number of frames at this point. And if you stare at these videos for long enough, you'll still see kind of issues that we don't want to be there, right? Sometimes the right paw marker hops over to the left paw, or sometimes when the animal kind of hides its paw back here, you'll lose tracking completely. You see kind of glitches in the data where the marker kind of jumps in a bad way, and that's going to spoil any downstream kind of temporal segmentation methods that we want to use. So we really need to try to make these methods work more robustly. That's still a pretty open problem. Any questions on this? Maybe some of you have used Deep Lab Cut or something similar and have experiences to share. I don't know. Any questions? All right.
It's a bit painful to use, but it's okay.
What's that?
It was a bit painful the time I used it. There was a lot of labeling. It was more.
Lots of labeling. Yeah, that's been it's been our experience as well. All right. So one of the things we've been very interested in is can we improve these methods, right? So one of the big trends in computer vision generally is moving from fully supervised methods to either unsupervised or semi-supervised methods, right? So Deep Lab Cut is a fully supervised method. You take a bunch of frames, you add your labels, you train your neural network just based on those labels, and then you hope that the neural network is smart enough at that point to handle all the other unlabeled frames in your data set. And sometimes it works great, and sometimes it doesn't work so well. So here's an example where it's not working so well. This is a pretty small labeled data set. We were trying to track these fingers. I think we have on the order of 100 or so labels on these fingers. So not very many, but you see that clearly that's just not enough, right? If you wanted to track these fingers over time, kind of every time the mouse moves a little bit, you kind of lose tracking. So this This just didn't work at all for us, at least without going to the effort of adding lots and lots of more labels. Okay, so you can do a few things here, right? You can think about active learning strategies where you kind of train a little, you label some data, you train a model, you look at the errors that the model is making, and then you try to correct those errors specifically, and then you train again, and you iterate back and forth until you end up with a good model, right? And that's kind of, I think that's a very good approach. It's kind of baked into the deep lab cut philosophy. And so that's one way you can improve these methods. A different way you can improve these methods is to pull in, other unsupervised or weak supervised signals, right? So what do we actually know about these videos, right? There's a huge amount of spatial and temporal kind of constraints that you can bake into these models and use them to train a stronger model, right? So for example, we know that the fingers on the paw are never going to be too far apart, right? We're never going to find a pinky finger down here and an index finger up here, right? That never happens. So one thing we can do as we're training our model is if it makes mistakes like that, if it puts the pinky finger down here and the index finger up here, we can impose a penalty that says, no, you shouldn't be doing that. You should learn not to do that over your training iterations. Bake that into the model so that it has this extra kind of unsupervised signal that it can use to learn hopefully a better tracker, right? So pay attention to the labels, right? Learn based on those labels, take gradient steps based on that supervised loss, but also have this unsupervised loss that says, okay, there are going to be constraints on how far apart these fingers can be, right? The other big constraint here is that the mouse's hand never teleports from one point to another, right? It always kind of moves smoothly, assuming that you have a high enough frame rate as part of your raw data, right? So you can impose a temporal constraint as well, right? Never let my thumb jump from one part of the screen to another from frame to frame. And if that happens, impose a penalty and train the model to do a better job, right? So if you stick in those spatial and temporal penalties as part of your training, you can train a stronger model. And that's what's shown here. This is a paper that came out, I guess, last year, showing that this simple approach works pretty well, actually. So this is using exactly the same training data set, but adding in these new unsupervised losses as part of the training. And you end up with tracking that is certainly not perfect, but it's significantly better than the supervised only training that we see above. All right. And then these other traces just correspond to, we're tracking the 1 finger, 2 finger, 3 finger, 4 finger down here. So you can kind of take a look at where these glitches happen in DLC and see if this new semi-supervised method actually corrects some of those glitches over time. And it turns out that it does. Any questions on this? Okay, so that's one way you can improve things. Yeah, go ahead.
That's unsupervised loss and supervised loss, and how are they combined in the training?
Sorry, this mic isn't working very well. I apologize. Can you repeat that?
Okay, there's this unsupervised loss and the supervised loss, and how are they combined during training? Is it like sequence or?
So yeah, that's a great question. So I'm going to skip the math here. What we used in this paper was kind of a stochastic variational inference approach to learn this model. So we basically have a latent variable that says, that corresponds to the pose that we're trying to track. So we have, in this case, it's going to be an 8-dimensional latent variable because we're tracking 4 fingers and we have the X and Y for each of those fingers. So we have an 8-dimensional latent for each frame. And then we impose, we have kind of a graphical model that imposes these constraints on each of the fingers and each of the relationships of the fingers from frame to frame. So we have a graphical model that imposes those constraints, and then we also pull in the information from each image, right? So the neural network is getting information from each image, and that further constrains these latent variables. And then we use, kind of pretty standard graphical model variational inference to actually train this model. So those details are described here. I think a lot of that can be simplified quite a bit. We don't need that heavy kind of kind of Bayesian machinery. So we're actually playing with simpler versions of this method now that will hopefully kind of be faster and easier to adapt to different needs. So we're hoping to release kind of an updated version of this code pretty soon. Did that answer your question?
Yeah, thanks.
Any other questions? Okay. Okay, so this is one kind of simple way of improving these methods. There are other things you can do, right? So I mentioned this active learning approach. So one thing that is very well known, if you look at the old school deep learning literature from decades ago, people realized very early on that if you train the same model with the same data set, but just randomize the order of the training steps that you take, you can get different models, right? These are wildly over-parameterized models that are caught in these weird local optima that we still don't really understand. So if you train an ensemble of neural networks, instead of just training a single neural network, you train 10 neural networks using either different hyperparameters or different training orders, you're going to get networks that make different mistakes. And those mistakes are going to be kind of idiosyncratic in a way we don't really understand very well. But we don't need to understand it. What we can do is just train those networks, and then, for example, take the median over the output of those different networks, and kind of for free, you get better tracking, right? So this is a very well-known phenomenon in the kind of old school deep learning literature. And of course, this is something that's trivial to do on AWS, right? It would be something you wouldn't want to do if you just had one local GPU that's going to kind of burn up training these 10 networks over and over and over again. But if you have access to 10 GPUs on the cloud, you can just do this kind of trivially. And it works, right? As you'd expect based on this decades old literature. Training an ensemble and using the ensemble to do the tracking actually leads to significantly more robust tracking with fewer glitches over time. The other cool thing you can do is use the ensemble to find bad frames, right? So if you look at where one network and one member of the ensemble and a different member of the ensemble disagree, that's actually a pretty good candidate for active learning, right? For putting in more supervised signals so that you teach these two networks to actually agree on those frames. So if you're doing one of these active learning loops, it's very useful to search for frames where two of your ensemble members are saying wildly different things. So we found that's pretty useful, and that can lead to actually faster training as well. All right. So this is kind of a simple old school idea. It's not very deep, no pun intended, but it's very useful, and it's kind of trivial to deploy if you're working in the cloud instead of kind of limited to your local machine. All right. So that's an easy one. What else can we do to improve here? Well, one of the questions that we've always had with these methods is, is it extracting all the information that we care about, right? So can we pull out more information from these videos that could be used to build richer encoding models or richer decoding models as we go into the brain and look at the neural activity? Or is it sufficient just to track the paw and track the nose and track the pupil diameter? Right. So that's a question we can ask directly, right? One thing you can do is run your favorite autoencoder on this raw data. Basically take raw frames, map those frames into a low-dimensional space, and train those low-dimensional latents to basically capture as much information in the raw data as possible, right? So that when you actually push these low-dimensional latents through a decoding network back into the original data space, you want the reconstructed frames here to match the original frames as well as possible, right? So PCA would be the kind of linear version of this, where you have a linear encoder and a linear decoder. Of course, you can do better if you swap in those linear mappings with nonlinear neural networks. And so this is something we looked at a couple years ago. Turns out to work very well. You can actually compress these videos down into a very low dimensional space, say 10 or so dimensions, and capture most of what's visible in these videos. So that's cool. But then we thought, okay, well, maybe these, can I do better if I have additional information, right? A lot of the games we play here come down to, what priors do I have? What do I know about the raw data that I can kind of build into my model to get better performance of whatever analysis method I'm thinking of. And so here, if we've gone to the trouble of labeling 10,000 frames and we've got trackers that work pretty well, why don't I pull that information in and use it to train a better kind of compression, right? So there are lots of different ways you could think about doing this. The way we've been pursuing recently is to use the supervised information, right? Use the tracker that we've already gone to the trouble of training to basically say, okay, I want to make sure that my encoder pulls out enough information to reconstruct the tracking, right? I still want to be able to do good tracking on these videos, but I want to get other stuff as well, right? So there might be, maybe the animal moves its shoulder in a funny way, in a way that's not captured by the movements of the paw, or maybe it wiggles its whiskers in a way that's not captured by the licking or the nose movement signal that we're tracking, right? So there's probably other stuff that's maybe a little bit orthogonal to these supervised signals that I'm tracking, and it'd be awesome if I could pull that out of the data as well, right? So you can make, I'm sure there are lots of different ways of doing this, but the way we did this is we made an encoder. Again, we're trying to take the very high dimensional raw video frames, map those into a lower dimensional space. And what we wanted this low dimensional space to do is to do kind of a couple different things simultaneously. We wanted to be able to push that low dimensional latent. We wanted to split that into two parts, right? There's a supervised part, which is responsible for having enough information to reconstruct the tracking, right? So we want to make sure we're able to do that. And then there's an unsupervised part that corresponds to kind of everything else we want to extract from the video, right? So we want to be able to push the supervised bits and the unsupervised bits together into a single decoder that actually capture the raw data, right? And we put together a couple loss functions that do this, and that's described in this paper that you can check out. Is that all pretty clear? What we're trying to do here and what the goals are? More or less? Okay. Cool. So now we can say, well, are we doing a good job, right? And so you can do standard things like look at how well you're able to reconstruct the frames as a function of the dimensionality of these terms. You can look at how well we extract the supervised tracking that we had before. What we really want to do, though, is take a look at these latents that we pull out and try to understand what they're actually encoding, right? So one thing that we found very useful is again kind of a classical technique from the autoencoder literature called latent traversals, right? So we want to take an image, extract its latent variables, and then hold those fixed and just change one of those at a time, and then push the resulting perturb latents back through your decoder and see what changes in the image, right? And if we're able to get kind of a good representation of a good low dimensional informative representation of these images, we should be able to kind of perturb these individual signals in a way that makes sense when we go back to the original image space, okay? So here's kind of an example of that. Apologies for this scary kind of zoomed in mouse eyeball here. But we took a single frame from our zoomed in camera. We're looking at obviously the zoomed in version of a mouse's face here. And then we're performing this latent traversal, right? So we're changing one latent at a time and just seeing what happens when we map that latent back into the image space. All right, so let's start with this one. This one's kind of the easiest one to think about. Here we know that we have supervised latents that correspond to the pupil size and position. So here we're just moving the latent that corresponds to the horizontal pupil location, and then when we map that back into the image space, we see this nice interpretable back and forth of the pupil. So that's working pretty well. Here's what happens if we change the vertical location. You can see the vertical pupil location is moving up and down a little bit. And then the obvious question is what happens if we look at the leftover stuff, right? What else is this model able to capture that wasn't in the original kind of tracked pupil supervised latents? So one of the latents, it turns out, corresponds to opening and closing of the eyelid, right? That's something that maybe we should have been tracking originally, but we weren't when we performed this analysis. Another one up here corresponds to horizontal wiggles of the whiskers. And I'm not showing it, but we also get another one correspond to kind of vertical motion of the whiskers. So if you kind of run this analysis, you can start to pull out things that are kind of pretty interpretable, and kind of you can separate these different signals in this low dimensional space into things that you can start to reason about and perform segmentation on or perform decoding, right? So here's an example of the latter, right? One of the things we're interested in is, can I take signals recorded in different parts of the brain and reconstruct this whole video, right? That would be a cool demonstration that we're capturing, important relevant signals that are reflected in the mouse's behavior, right? So here again is kind of a chunk of the raw video data. What we're doing is running this fancy semi-supervised autoencoder. We're extracting a low-dimensional representation and then mapping this back into the image space. And what you see is that it's working pretty well, right? We're certainly missing some of the high-frequency kind of pixely level stuff, but we seem to be capturing most of what's going on in this video, in this reconstruction. So we're pretty happy with that. And then we can go ahead and take signals from primary visual cortex or motor cortex or elsewhere in the brain and ask, okay, how well can we actually decode the low dimensional representation that's being displayed here? And that's exactly what we did. So in this case, we took some signals from visual cortex, tried to build a decoder that tracks these low dimensional latents, and then mapped those latents back into the image space to see how well we're doing. And if you stare at these videos for a while, you see that there's a lot of whisker wiggles that maybe unsurprisingly are not really captured by the visual cortex reconstruction here. We're able to do a pretty good job with some of these latents, but some of the latents are just kind of missing completely in this, at least in this small population of visual cortical neurons that we're looking at in these trials. Is that all good? Any questions on that? All right. Okay. What are some other games you can play here? I think one of the big unsolved problems here is temporal segmentation, right? So after you've extracted out this hopefully rich and low dimensional representation of the video behavior, you'd like to do things like understand, okay, the mouse is reaching now, or the mouse is resting, or the mouse is scrubbing its face, or something else, right? These mice do lots of different things, and it's we're guessing that these different behavioral states are going to correspond to different neural states, and we'd like to correlate back and forth between the two. So to do that, we'd love to be able to kind of perform temporal segmentation on these extracted signals, right? And so again, there's been a huge amount of work doing, fully unsupervised methods where maybe you fit kind of a hidden Markov model to the temporal features that you pull out of these videos. On the other side, there's fully supervised methods where you label a bunch of segments of video, and then you train your favorite neural network to perform segmentation. Again, we're kind of interested in combining the best of both worlds. So how do we come up with an architecture that allows for unsupervised information and supervised information? So this is a short paper that's on the bioRxiv. You can check it out. In this case, we have 3 different losses that we've added. There's a fully supervised loss where we've labeled a few segments of behavior. Most of the data is not labeled because we're not patient enough to go through the whole data set to do the labeling. So we only have kind of a subset of frames that are kind of labeled here. But we also have a few other kind of simpler heuristics that we can add to the data, right? So maybe we say, well, I think that usually when the paw is really close to the face, maybe that's kind of grooming behavior, right? So I'm gonna say I'm not sure about it, but I can certainly measure the distance between the paw and the face, and if that distance is small enough, I'm gonna say, okay, maybe that's grooming behavior. All right, and because that's a cheap thing that I can do without having to hand label lots of different frames, that's a signal that I can apply to the whole data set, right? So that's kind of a weak supervision signal. It's not perfect, but it's probably correlated with the grooming kind of states that we want to extract. And then we can also do things like, okay, we want, whatever representation we're pulling out of this this video stream, we want it to be informative, right? We want it to capture a lot of information in the raw data. So maybe what we can do is do some kind of auto-encoding, right? Make sure that whatever the kind of low dimensional space we end up working in, whatever that space is that we learn, we want to make sure that it has good predictive qualities, right? We want to make sure that we can start from that low dimensional representation and then, for example, predict the behavior in the next time frame, right? And that's a fully, it's like PCA again, right? It's a fully unsupervised method that we can run on all the data without doing any labeling. And the hope is that when we combine this unsupervised loss function with the semi-supervised loss functions corresponding to these heuristics of what these behaviors should look like, along with the strong supervised signals of, okay, I know on that segment, I know exactly what the map is doing and I've got it hand labeled. Combine these three different losses, the hope is that we get better tracking. And so far, the result is that's the case, right? If you can weigh these different signals properly, you can end up with very good reconstructions of still versus walking versus front grooming versus resting and so on. And so these methods seem to be pretty promising, and we're trying to kind of roll this out on more data sets now. So that's work that's still in progress. All right, so the hope is, hopefully you've seen there's a huge amount of information that you can pull out of these videos. I think we're actually still in kind of the early days of methods for handling this data and kind of making it work at large scale with as little kind of user input as possible. I do think that user input is very important, but we want to minimize user effort. We're happy to let computers do as much of our work for us as possible. And so I think these semi-supervised approaches that I've talked about here are kind of a good path forward for doing some of that. Any questions on the behavior stuff before I move on?
So, Ryan, I have a question on the semi-supervised VAEs, if that's possible.
I didn't quite hear that. Can you try again? Sorry.
Something before the semi-supervised autoencoders, if that's possible. Still to ask you.
Yeah, I'm sorry. Maybe put the question in the chat and I can respond to it there. I'm sorry, I'm finding it hard to.
Can you hear me?
Yeah. Okay.
I had a question on the semi-supervised autoencoders, if it's possible.
Yeah.
The latents could, in general, be any combination of the two interpretable latents that you showed. How do you reach easy to interpret latents? I mean, is there a way to automate this?
Yeah. Great question. Yeah, I should have been more clear on that. So I showed you the architecture, but not the loss functions here. So we have several loss functions that are added together as usual. We have the reconstruction loss, right? So we want to make sure that we can match the tracking performance that we had before. We've got this reconstruction loss, so we want to make sure that the output here is as close as possible to the raw data as possible. And the thing I didn't mention, which is important, which I should add to the slide, is an unsupervised loss where we try to decorrelate these latents as much as possible, right? So we have kind of, people have tried lots of different ways of disentangling these unsupervised latents from each other. So you end up with something that's, think of it as doing like independent components analysis, right? We want each of these latents to be as independent from each other as possible. And so we took one of those standard losses from the literature and just added that in here. So think of it as kind of an ICA loss. And yeah, that's what led to, this kind of thing where kind of the motion of the whiskers tends not to be completely correlated with the eye blinks. And so you can pull out a couple different kind of more or less disentangled signals here with this loss. So that's a great question. I should have been more clear about that.
Thanks a lot.
Yeah, thank you. Any other questions?
Yeah, in the behavioral stuff, you had to set some temporal scale, right, for the behaviors, right? And I mentioned like but the whisking is much faster than for like an arm movement or something. But
That's right.
Yeah, maybe you could mention something on that.
Yeah, that's a great question. So, and I think that's kind of a weakness of these semi-supervised methods, right? You have these different lambdas, the different kind of weights that you're going to add to the supervised term versus the unsupervised term versus the different kind of losses that you have here. So some of those can be set kind of automatically. So let me go back to the this deep graph pose stuff. So yeah, so in this case we have a couple different additional losses that aren't in the fully supervised setting. So we have these spatial terms and also these temporal terms. So one thing you can do here is use what's called an epsilon insensitive loss, right? So I'm not going to worry about as long as the thumb and the pinky are close enough together, as long as they're within some epsilon, I'm not going to worry about it, right? And that epsilon can be set automatically, right? If you label a bunch of data and the thumb and the pinky are never further apart than 10 pixels, then you can set epsilon to be 20 pixels and be done with it, right? So some of those losses or parameters can be set automatically. But other losses, you might have to kind of play around with a few different hyperparameters until you're happy with the results. Some of them, maybe you run cross-validation to kind of pick those hyperparameters automatically. It's definitely kind of, that's definitely a drawback of these fancier methods. You do end up with more hyperparameters to think about. So we didn't do it in the original paper, but I think moving forward the approach there is again just to run the hyperparameter search in parallel over multiple instances on AWS to try to minimize the user effort. So let the computer handle as much of that hyperparameter search as possible, and again that's much easier to do, much more tractable to do on the cloud than it is on your local heated up GPU. Other questions? All right, awesome. So let me jump ahead to the last topic I wanted to talk about, which is this the question of multi-electrode data, right? So obviously, a lot of you probably have experience with these neural pixel recordings. The IBL is, our first pass at this brain-wide map is going to be completely based on these neural pixel recordings. If you haven't seen them before, they're kind of a couple things that are important to know about. So these are kind of long linear kind of probes that have many electrodes kind of embedded along the probe, right? And because of their size relative to the mouse brain, you can't help but get recordings from multiple brain areas simultaneously here, right? So in this case, this probe goes through multiple brain areas, the cortex, the hippocampus, down into kind of a variety of subcortical kind of regions. And so you end up with data that looks like this. It's kind of localized along one dimension, the length along this probe, and you can, on a good day, you can have, clearly separated signals from, hundreds of cells in multiple brain areas. So it's an extremely rich source of data, and obviously, these techniques have now been rolled out to hundreds of labs around the world. So this is really a huge source of data that that we need to do a good job on, right? So this is a pretty important challenge for us as a field to develop the platforms that can handle this data and develop good analysis methods that build on these data sets. Okay. So given that I've gone a little bit long already, I'm gonna skip some of the details here, and really just focus on NeuroPixels recordings. I could give a whole talk on other types of ePhys data, but let's focus our attention on NeuroPixels recordings for this talk. Okay, so one of the really great things about this data is because these electrodes are pretty densely spaced, you can record simultaneously on multiple channels. You're going to be able to sense the signals of each cell on many channels. typically on the order of 10 or so channels. And so what this buys you is you have a lot more information about each spike that you can use to perform spike sorting. So here's this is actually some retinal data, but NeuroPixels data looks very similar. If you look at 7 electrodes worth of data over just 10 milliseconds, it's extremely rich data. If you just look at this data, you see, okay, here's a big spike that happens here. Here's maybe a smaller spike that happens here. Here's maybe a different small spike that happens there. The goal of spike sorting, of course, is to take this raw data, detect all these little spikes that happen, and then assign each of those spikes to individual cells, right? Perform some kind of clustering, some kind of demixing, just like we talked about in the calcium imaging setting, to go from the raw data to a representation of neurons by time instead of channels by time. All right, and the thing you should know about spike sorting is it's very much not a solved problem yet, right? So there have been a lot of papers that have kind of been written on the spike sorting problem, but you should not trust any of them. Some of them work pretty well, some of them don't work very well, and just when you're performing your analyses and your cool theoretical models based on these NeuroPixels recordings, just be careful, right? I'm going to talk about some of these issues as I go, but that's kind of the bad news. The good news is that, again, this is extremely rich data, and you can, if you're doing a good job of spike sorting, you can actually assign even little small blips in the data that, if I was just recording from this electrode, I would have no chance of knowing that this was kind of a nice little spike that I'm seeing there. But if you have this full spatial temporal context from multiple channels, you can start to do things like, well, okay, I saw that this cell spiked here, and I can actually infer that, okay, that's that little blip of activity. That's not really noise. That's the activity of a well-defined cell that I can correlate back to this original electrode down here. So you can actually start to assign very small signals that, because they're happening synchronously with other signals that are much larger elsewhere on the array, you can start to perform this demixing in a much more accurate way than was possible if you just had kind of single electrodes or kind of a small handful of electrodes. Okay. All right. So, like I said, let me skip over. We've done a lot of work in trying to push these spike sorting methods forward in the context of retinal recordings. I think I'm going to skip over those in the interest of time. You can. I'd be happy to talk about those offline if you're interested. But let me kind of focus for the last few minutes here on NeuroPixels recordings. Okay. So again, as part of the IBL, we've been very interested in rolling out existing spike sorting code on hundreds of data sets. And it hasn't gone well. Sometimes the code just fails for no apparent reason on some data sets. We see all kinds of artifacts that are not really emphasized in the literature. So you see things like this, for example. So let me kind of orient you to what we're looking at. Here, it's just a rastergram of multiple cells recorded on a single array. All right, so here's time down here. Here's space up here. This is kind of the depth along this linear probe. And what we've seen a lot of are these weird stripes, right? So where all of a sudden, the whole brain basically will be kind of firing simultaneously. And maybe this is something cool that's going on in the whole brain, and it's kind of corresponds to communication between multiple brain areas. Or maybe it's just an artifact, and you didn't kind of pull that out of your raw data very well. In this case, we actually think it's an artifact. If you go back and look at the raw data, you see these big synchronous kind of noise events that are get pushed through your spike sorter, and you end up with spikes that are completely artifactual. All right, so that's pretty bad news, and we've been working on doing stripe subtraction in the raw data to kind of pull that out. Another big issue, you see these stripes again in this data set over here. This is just a different data set from a different mouse, but you also see drift, right? So this cell is nicely localized going along, and then all of a sudden, the electrode array might have shifted very slightly with respect to the rest of the brain. Right. And if your spike sorter isn't capable of kind of tracking these non-stationarities over time, any kind of downstream analysis you do based on the correlation between these different cells is going to be completely swamped by this very large kind of non-stationarity that's in your data. So you have to be very, very careful about that. One kind of interesting thing about this drift is that it's actually spatially non-stationary as well. So if you look down here, kind of deeper in the brain, sorry, I guess this plot is flipped. So this is more shallow. So there wasn't much of a drift relative to the brain down here, but there is a huge shift up here, right? So we need methods that can handle that properly. And then on some data sets, there's just not as much going on in some parts of the brain as we'd expect. So that makes us worry that maybe something weird is going on in those recordings, right? So to be clear, I don't think this is an issue for IBL data per se, right? These are some of the the most skilled kind of experimental labs in the world who kind of developed these neuropixels array recordings originally. But the difference, I think, is that we're looking at this data at very large scale. We have a lot of people looking at the data and trying to do multiple different analyses on this data. And so we've discovered a lot of these artifactual issues that I think are probably present in all NeuroPixels recordings. And they're just not getting noticed because fewer people are looking at some of these data sets. All right, so that's kind of the bad news. So we've been working on ways of improving things, and I should say, there's lots of people working on ways of improving these recordings. Here's something we've been focused on. We can try to look at these wobbly data sets over time and try to develop more robust methods for kind of registering the data, right? So one obvious thing you can do if you look at these, here's a data set where the experimentalists actually kind of push this probe up and down over time, so you have kind of you know what the wobble should look like. So it's a nice data set for testing out these registration methods. So if you just perform kind of image registration on these multiple kind of frames of data, you can start to kind of lock down this wobble in a way that it's much more stable over time. So that's something we've worked on and other groups have worked on as well. I think there's still room to improve here. Another big thing that we've worked on more recently is trying to pull out a more efficient representation of the data. So what do I mean by that? So what I'm showing here is these orange markers correspond to the locations of the array in space. So in this recording, this is a NeuroPixels 2.0 array. So you have kind of just two columns of electrodes going in this long line across a large part of the brain. So we're just showing kind of a subset of the electrodes here. And if you take each spike that you detect on these multiple electrodes and denoise the data a little bit, it turns out you can do a pretty good job of triangulating the location of each of these spikes. So you can kind of treat this big electrode array as a big kind of antenna array and look at the magnitude of each of these signals and each of these electrodes. And if you make a simple kind of point neuron, very basic, incomplete, flawed model of the biophysics here, you can write some code to do triangulation of these events. And what we're plotting here is the output of these methods. So this yellow clump of cells corresponds to a bunch of spikes that are very close to the electrode array, where the color here corresponds to the size of the spikes. So big spikes tend to be very close to the array. Small spikes out here tend to be much further away and more poorly localized, right? So smaller spikes are going to be noisier, and it's going to be harder to localize those in space. But if you look at this, what you see is that a lot of these little nice clusters in space can be very well localized, and you can use those as features for downstream clustering or registration of the data. So this is a very informative kind of feature that we can pull out of the data and use that for downstream tasks. So here's a little example of that. Again, we're looking at the electrode array here. Now we've changed the colors. I guess we should have kept the old color scheme, but we've changed the colors. Each dot here corresponds to a spike. Each green dot here corresponds to an electrode on this array, and in this case we're looking kind of face on the electrode array, where we're performing a three-dimensional kind of a max project onto the data. Over here we're looking kind of with the array kind of sideways, So basically, the purple spikes down here are kind of far from the array. The yellow spikes up here are close to the array. And we can see very clearly that, in this wobbly data set, we can see these clusters, and we can see how they're wobbling over time. And we can use this wobble to kind of, try to register the data over time. So that's what's being shown in the bottom row down here, where we're taking kind of a denoise representation of these clusters of spikes over time, and then we're performing a registration to try to make these clusters stay as kind of still as possible over time. So that we can then cluster them and run our favorite spike sorting tools on the registered data. Is that all clear?
I have a question about the wobble. So if I understand correctly, the wobble is a way of perturbing the data and trying to correct it, right? And trying to learn how to fix that in a way. So it's a wobble that's intentionally done by the experimenter to be corrected later.
Exactly. Yeah. So this was all right. So we see drift in a non-trivial fraction of recordings. Maybe in, say, a third of recordings, you can kind of visibly see some drift over time. And of course, there's probably many more recordings where if you zoom in further, you could probably detect drift as well. In this data set, and so that's just kind of natural drift. Just we tried our best to keep the electrodes still over time, but the animal moved, and maybe there was some slight drift in the electrode position over time. In this data set, this was kind of purposely recorded to test the limits of drift correction. So they imposed. I mean, you can see the locations of these cells drift quite a lot with respect to the electrode array, and you can see large changes in the shape of these spikes over time. But of course, the advantage here is that we know what drift was injected. If you look at this data set, there's actually quite a lot of natural drift as well, kind of ironically. There's a combination of kind of slow drift that you can see over part of the recording, and then this bigger drift that you can see that's experimentally imposed. And so we can kind of try to track both of those over time here.
How similar is this to, I mean, the artificial drift, how similar is it to natural one? I mean, the natural drift also goes both ways.
Yeah, that's a great question. So there are a couple differences. So this drift, it's periodic in a way that you don't see in real data. The scale was chosen to be pretty generous. So this drift is larger than we usually see in real data, but we've certainly seen data sets that have this size drift or even bigger. So the scale is maybe on the generous side, but certainly comparable to real data sets. And the speed is also chosen to kind of match real drift that we see in data. So I think the big differences are that real drift isn't constant and periodic in this way. It's in real data you typically see kind of more or less stationary data, and then a little bit of drift, and then more or less stationary, and then maybe a little slow drift over time. But in other respects, it's pretty well matched to real data examples that we've seen.
Thanks.
All right. Other question?
Is it not sufficient just to normalize the data and get it zero? Or just
I didn't hear that one. Apologies.
Is it not sufficient just to normalize the data, so it will get rid of the low frequencies so the mean stays constant?
Did you hear? The question is it not sufficient to get rid of the mean and basically the low frequency?
Yeah, great question. So yeah, I should say, I think there's a lot more work to be done here. So I would love to hear any of your clever ideas for doing this in a simpler, better way. Yeah, so if you're able to cluster each of these things, so the problem is that we don't know the identity of these spikes a priori, right? We just collect a bunch of spikes and we can extract an estimate, a crude location of the estimate of each of these spikes, but we don't actually know we haven't done clustering on these spikes, right? You could do a lot of spike sorting on each of these little frames and then try to match up each of the clusters that you get on frame 1 with frame 2 and frame 3 and so on, where the frames are, maybe a second worth of data or so. that's expensive, and any errors you make in that clustering step are going to propagate into your registration step. So we've tried to use methods that don't rely on clustering, that just try to take this raw data representation and try to register one frame to another. So yeah, if you knew exactly where each of these clusters were, you could kind of take means and then just subtract off the mean. But you have to believe those means, I guess. And if you don't believe those means, then you might actually end up injecting more noise into your recordings than you had originally. Also, you have to be a little bit careful about this non-stationarity over space, right? Some areas move a lot more than others, and so you wouldn't want to just extract a single mean across the whole array and apply that. You want to kind of operate a little more locally, both in space and in time. Did that address the question?
Yeah, thank you.
Great. Okay. Yeah, so that's most of what I wanted to say related to this, the spike sorting stuff. Obviously, I've purposely, there's all kinds of cool stuff you can do with spike sorted data, right? Most of my career has been spent kind of avoiding spike sorting and playing fun games with, building cool models of the correlations between cells or decoding spike sorted neural activity. But I think we really have to stop and address these spike sorting questions now, because we want to have kind of a nice solid foundation for all the analyses we want to do downstream. And so there's a bunch of kind of open questions here. If you guys are looking for course projects, I'd be happy to discuss any of these further. There's lots more work to be done here. And so yeah, I'd welcome further thoughts or input here. I guess I'll close on this last slide. I think over the last 10 years, like I said, it's been a super exciting area to work in. It's really crazy to think about where we were 10 years ago and where we are today, how far we've come. But I guess my overall feeling is that there's still kind of a huge amount of work left to be done, and we really need young, clever scientists to be kind of working in this area and pushing things forward. I guess the big kind of meta point that I tried to emphasize throughout here is this kind of scaling gap. So going from kind of a single data set, methods that work well on kind of a nice single data set often break when you kind of go to scale. And I think that's going to be an issue for the whole field as we see more and more of these multi-lab collaborations kind of produce very large, interesting data sets. And so I think that's a big issue for the field that we need to grapple with. And I guess you could tell from my last set of slides that my overall feeling is that there's been a decade's worth of work on calcium imaging data. There's been obviously multiple decades worth of work on computer vision that we've pulled into behavior video analysis that's made a big impact. I really think that NeuroPixels recordings are being done, like I said, in hundreds of labs now. And if I had to pick kind of one of these data sets where I'm kind of more nervous about the basic data quality, I really think we need to work harder on NeuroPixels recordings. It's going to be a hugely powerful platform, but we really need to work as a field to improve the basic processing chain there. So I think that's really important to remember as you go ahead and try to analyze some of this data in your future work. So that's it for me. Thanks so much for all of your great questions.